Now, as I promised at the end of chapter 3, we are continuing our discussion of patterns of inheritance with consideration of traits that do not follow simple Mendelian patterns of inheritance, or at least expression in the phenotype of the offspring. Here you can see a group of puppies that obviously are providing evidence of multiple different phenotypes that are possible, and even within those that are black and white, one of the three clear types of puppies that you can see, it's pretty obvious that not all of those are equally black and white. And so, how is this phenotype derived if there are only two alleles, two copies of the gene present in each of these puppies? So, what's going on? We're going to dig into this, and first we need to start with where the field went after Mendel's work was rediscovered and its importance recognized in the context of inheritance of traits and evolution of species. So, after Mendel, researchers focused on traits that didn't follow these simple patterns like those illustrated in Mendel's pea plant experiments, right? He had basically explained how that works. So, everyone who was working in the field at this time was left trying to figure out the odd things that don't follow those patterns. And it turns out there are a lot of traits like this. So the result of this work was the understanding that the patterns of inheritance are the same as Mendel described, but the traits might be influenced by other things. Okay, they might be influenced by more than one gene present in the genome. They might be influenced by the environment. They might be influenced by the gender or biological sex of the individual. And so these are the things we're going to dig into. Now, you understand what an allele is. It's basically a copy or a version of a gene, right? And most genes, have multiple versions that exist within a species. For example, the gene that controls the color of your hair within the human species, everybody has two copies of that gene, and there are a number of different versions of that gene, which we call alleles. And it's important that we understand that when we're talking about genetic variation within species, these different versions of the gene that exist within the species are referred to as alleles, and that's something that you understand already. But now I'm going to introduce a few more concepts to you. The first of which is the idea of there being a wild type. And a wild type, that term is used to refer to the allele that is most common in the population. All right. So within the species that I've illustrated here, leopard geckos, the wild type coloration is what you see here. They have kind of a yellow body with a purplish white tail and black spots all over them. That is the wild type phenotype. But as people have kept these, they make fantastic pets, and bred them, they have selected for specific traits, rare alleles, and sought to increase the frequency of those rare alleles within the captive population, selectively breeding individuals that have those rare alleles, like the mutant allele that produces this bright orange coloration. And so now you can purchase a leopard gecko. You can choose one that has the wild type phenotype, or you can purchase a leopard gecko that has this bright orange coloration that is due to the presence of mutant versions of the gene, so alleles that are the result of mutation. Every allele that exists is the result of a mutation to an existing gene copy within the population. So all of the variation that exists within species is the result of mutation. The most common version of the gene, most common allele in the population, is referred to as the wild type allele, and the phenotype that is driven by the expression of that wild type allele is the wild type phenotype. So you might think of a number of things that you might see while you're out outdoors looking at, let's say you go to the zoo and you see two tigers, or you're watching Tiger King and you see two tigers. One of them is the normal orange with black stripes. That's the wild type phenotype conferred by the alleles that cause the expression of that orangish coloration. Conversely, you might also see white tigers. They still have black stripes, but instead of on an orange background, it's on a white background. That is a mutation that has affected the expression of coloration, the color genes in those tigers, and therefore their background color is white. Okay? So the wild type would be the normal orange and black tiger, and any other version, not just the white ones, but all the others, are the result of mutations to that wild type allele. Now, I spoke to you in the previous chapter about notation methods utilized for denoting different alleles for a gene. And in that chapter, we used primarily capital and lowercase letters. All right. And the capitalization generally referred to an allele that was dominant, and its phenotype would be expressed at the expense of the recessive allele. But as we're going to learn, that is not always the case. There is not always a dominant and a recessive allele in a particular gene system. And for that reason, we do use other means of expressing alleles that reflect whether they are the wild type or simply expressing their differences from other alleles. And so, again, the letters generally come from the name of the mutant. So the example I give here is a little e, and that is a mutant allele for a gene that controls whether or not fruit flies have eyes. So the little e might refer to or be the result of calling the phenotype eyeless. So they use a little e to refer to the eyeless allele and the eyeless phenotype. Okay, so there's another example down here, WG, where that might illustrate an allele, a dominant allele for a particular phenotype. And this is a phenotype whether the fruit flies have wings or not. Okay, and the WG is the dominant form. They would have wings, but the lowercase wg means they are wingless. Okay, so just realize that just because I give you an allelic combination or an allelic notation, whether it's two little e's or a big E and a little e or two lowercase wg's, you don't necessarily know right off the bat that you are able to infer the phenotype of that individual unless I tell you something about the expression of those alleles, whether one allele is dominant to the other, etc. Referring back to the new term I've introduced to you, wild type, frequently a wild type allele will be indicated by a superscript plus. So in a particular genotype, you might see E plus, so they're homozygous for the wild type allele, or just EE with no plus sign. They're homozygous for a mutant allele, or heterozygous would be E plus and E. All right? So you may see these different notations in the textbook and the problems at the end of the chapter, which are always useful, and in some of the online materials that I'll have you working through. Just recognize that you can't always assume, unless you're told, that a particular allele, just because it is capitalized, is dominant. Okay? In some cases, as you'll see in a moment, the different alleles are all capitalized. So there are some instances where the alleles that exist within a population do not have a simple dominant recessive relationship to one another. Okay? And in this case, the letters from the gene name might be indicated and followed by some superscript that simply denotes the different alleles. So for gene R here, there might be two alleles, R1 and R2. Or gene L, there's LN and LM. Two different alleles for gene L or gene R. And those alleles, this notation, gives you no indication of dominance or recessiveness. They are simply the two alleles that exist within the population. So not everything has a dominant or recessive relationship when it comes to the expression of different copies of genes within an individual. So let's look at some of these examples and how that works. Now, we're going to work through some systems, and it's important that you understand and are able to differentiate these types of patterns of inheritance. This first one being incomplete dominance, okay? We're going to look at one in a moment that's called codominance. They're very different. It's important that you understand the difference. So in a system of incomplete dominance, we have multiple alleles. We're going to work with a simple case of two alleles. But in this instance, neither of those alleles is dominant to the other. So let's look at some flowers whose flower color is controlled by a gene that, when expressed, results in the coloration being either red or white. So there are two different alleles. Okay, we'll just call them R1 and R2. This top left red individual, okay, we're doing a monohybrid cross here. It's a true breeding red individual. That means that individual is homozygous for R1, crossed with a true breeding white individual. That individual is homozygous for R2. Now, what's the genotype of all the offspring? You know this from our previous chapter. All of those F1 offspring are going to be heterozygous. Their genotype, heterozygotes, is R1, R2. They have one copy of R1 from the red parent, one copy of R2 from the white parent. So they've got one of each. And what we see is that these F1 offspring are 100% pink. All of them are going to be pink. They all have a red allele. They all have a white allele. Well, what if you express red and white simultaneously in every cell of these flowers? You don't get a red flower, you don't get a white flower, you get a pink flower because of this incomplete dominance. Both alleles are expressing themselves. You're getting the red and the white, and it's mixing, and you get a third phenotype. If we cross these F1s to create an F2 generation, just as we did in the monohybrid cross, you'll see that we get one red, two pink, and one white. Now, this is only showing 4 offspring, but if we had 400 or 4,000 or 4 million, the ratios would hold in that we're going to have 1 red for every 2 pink for every 1 white. Now, if you think back to the genotypic and phenotypic ratios we described in chapter 3 with Mendelian inheritance of these pea plant traits, you'll remember that our F2 generation consisted of a ratio of 1 homozygous dominant individual for every 2 heterozygotes for every 1 homozygous recessive individual. What we have here in this F2 is we have the same genotypic frequency. 1 R1 homozygote for every 2 heterozygotes for every 1 R2 homozygote. Okay? So this genotypic ratio is exactly the same as these simple dominance examples that we worked through in chapter 3 with Mendel's pea plants. But what we're seeing here is that unlike those examples, the phenotypes of the F2 generation, there are 3 of them instead of 2. Remember, in the pea plant experiments, the homozygous dominant individual and the heterozygotes had the exact same phenotype. Here, those two, the red and the pink, have different phenotypes. And so when you have a monohybrid cross that results in a new phenotype in the F1 generation and 3 different phenotypes in the F2s, we've got a system of incomplete dominance, or potentially what I'm going to talk about on the next slide, codominance. So codominance is very similar to incomplete dominance. Okay? In codominance, the influence of both of the alleles is apparent. Okay? But what's interesting is it's distinctly expressed independent of the other allele. So What I mean by that is, unlike the flower example, where when they have a red allele and a white allele, you get pink, what you would have in codominance is you would have some parts of the flower be red and some parts of the flower be white. I encourage you to Google codominance and flowers, and you'll see what I'm talking about. You get a very different result than you did in the previous slide. Okay? So, a great example that we're going to use for codominance is that of human blood type. Okay? And in this, the influence of both alleles is apparent. So you get joint expression of these alleles. Okay? So if you're homozygous for one allele, your phenotype is that allele's phenotype. But if you are heterozygous, your phenotype is both of those things. And so what we're seeing is distinct expression of both of these alleles and not a blending. So let's use a more familiar terminology or more familiar letters. We'll talk about this in the context of being type A, type B, or type O blood, or possibly even AB. And so what we're seeing here is two important concepts. We are seeing the illustration of codominance, but we are also seeing that, unlike most systems we've talked about so far, in human blood type, there aren't two alleles. There are three alleles for this gene. And each of your parents could give you, they can only give you what they've got, but you could receive from your parents an A allele, a B allele, or an O allele. All of these alleles exist in the population. And this illustrates an important point in that there are many, many alleles for many genes, okay, in humans as well as other species. These traits are not limited to having one mutation that creates a second allele, and that's it. So in human blood type, we have three different alleles, and the phenotype that an individual has is the result of expression of both copies of the gene that they possess, received from different parents. So if you are type A blood, the first column there in this table, okay, you have on your blood cells, you have an A antigen sticking off the surface of your blood cell. That's those kind of pink lollipops here that are sticking off the surface of this A blood cell. What that means is those antigens identify that blood cell as being type A. And this individual that is type A will also produce these antibodies that are floating around the bloodstream that work against type B blood. They will bind to, identify, and fight type B blood. That's why if you're type A, you don't want to receive an infusion of blood that is not the same type as yours, because those antibodies will attack them and cause you serious problems. Similarly, type B blood, they have B antigens on the surface and they produce A antibodies. Now, if you are AB, if you are heterozygous, if you received a type A allele from one parent and a type B allele from the other parent, your blood cells have both antigens on the surface. And necessarily, you can't have any antibodies present. You'd be fighting every one of your own blood cells with them, okay? And then type O, they have a unique type of mutation that we'll talk about later, in that they have no antigens on the surface of their blood cells, as you can see here. So their blood cells do not identify as being either A or B or AB. But they do have antibodies to both A and B. And so I want you to look here and think about this for a second and think about why type O blood, those individuals are universal what? Donors or universal recipients of blood? Group O blood type, those individuals are universal donors. If you and I are each AB and the other person that's watching this lecture is type AB, all three of us could receive type O blood. And we wouldn't have any problems, because even though two of us have antibodies present, they don't identify type O blood as being any different than what we've got normally. So we can receive type O blood. AB is the universal recipient. They can receive any blood. They can receive A, they can receive B, they can receive O. Doesn't matter, okay? Because they have no antibodies present. And so in this system, for these alleles, we would denote them as being, for example, I with a superscript A, a superscript B, or a superscript O, okay? Now, to reiterate the fact that we can have many different alleles for the same trait, here's just an example of a fruit fly, a model organism used by many geneticists, and the many different eye colors that these species have. This just illustrates a few, but just like humans, there are lots of possible eye colors, and these are controlled or produced by different mutations to the same gene. So many alleles for the same locus that can result in different phenotypes. All right, now, as we consider the types of mutations that might occur, it's not surprising to realize that some mutations could prove fatal, okay? Mutations occur, and we have a whole chapter on mutations. They are random changes to the genome, random changes in the genetic code. And if you think about that randomness, it's quite possible that the random nature of those changes could be very detrimental to the health of an individual. In many cases, they might result in that individual not even being conceived or not even proceeding far beyond conception, okay? So some of these mutations can be incredibly deadly. However, they might only be deadly if an individual is homozygous. You might be able to survive as a heterozygote with a lethal allele, okay? So some mutation that is so bad that it completely inactivates the function of that locus. So the thing that is produced by expression of that gene is useless and potentially fatal, could be countered by the fact that the individual has a functional copy they received from their other parent, okay? So how might lethals persist in the population? Because it seems like it would be a bad thing. We might observe lethals within a population, much as were observed by scientists when they crossed these mice that had these two different coat colors, agouti, that sort of dark brown color, or yellow coloration of the mouse on the bottom there. When they crossed these mice, they observed that when you cross an agouti with an agouti, you get all agouti. So this is a true breeding genotype, and all the offspring have the same genotype, okay? Now, if you cross a yellow with a yellow, you would always observe 2/3 yellow, 1/3 agouti offspring, okay? And then crossing an agouti with a yellow, you would always get half yellow, half agouti. Now, this is a cross I encourage you to work through, because I haven't told you the genotype of these individuals. I'm only telling you their phenotype. And it's a useful thought exercise to work through how this could possibly work. And the real key to this is the last two crosses. If you breed these any yellow mouse that you have, you get these unusual results that you would not predict if you do the Punnett square and work it out, okay? And the key here is that Y is dominant, but it's lethal in homozygotes, okay? So just take that. I didn't type the fact that it's only lethal in homozygotes, but I want you to work through the Punnett squares and see why you get that result in the middle, why you get 2/3 yellow and 1/3 agouti when you cross two yellow mice, okay? So if it's lethal in homozygotes, What is the genotype of those two yellow individuals in that middle cross there? If you know that, do the Punnett square, and you will see why you get this unusual outcome that might seem to defy the expectations based on Mendelian patterns of inheritance. Now, what's perhaps even more surprising is how dominant lethals might persist in a population. I mean, if a mutation kills you, you would expect selection to remove it from the population. It should disappear, it should be selected out. But you might be surprised to learn that some of the diseases that you've probably heard of, particularly here, Huntington's disease, this is a disease that is the result of a lethal mutation. So a dominant lethal mutation to a gene that results in 100% mortality. The reason it sticks around in the population and has not disappeared because everyone that has it dies is because the effects of this mutation do not manifest themselves. The individual does not pass away until after their reproductive years. And so it doesn't influence the passing of this allele on, its continuation within the population. All right. So now that we've spoken about the multiple varieties of alleles that can exist for a particular gene, I also want you to start thinking about the fact that we have thousands of genes, but only tens of chromosomes. And what that should immediately lead you to realize is that many of your genes are physically connected to one another in that they exist on the same chromosome. Here you can see a terrible cartoon of a replicated chromosome, each of these halves being sister chromatids, and they should bear the exact same genes, and they should bear identical copies because they are sister chromatids. Now, what I want you to think about from this figure is that the genes that occur on the same chromosome, and they're close together even, they will have alleles that exhibit what we call genetic linkage. Okay? Because they are physically connected to one another, they are passed from parent to offspring together. Now, you will remember that important point I spoke to you about in an earlier chapter, independent assortment, that being the whichever parental copy of chromosome 1 you got from your mother. So your mother has grandma's number 1 and grandpa's number 1 chromosome. She only gave you one of those, okay? But that sorting, that segregation in meiosis 1 occurred independently of whether she gave you grandma's number 2 chromosome or grandpa's number 2 chromosome. So that independent assortment lets mom mix up what she gives you. A little bit of grandma, a little bit of grandpa, okay? But on each of those chromosomes, because there are so many genes on every chromosome, let's say chromosome number 1 has the gene for eye color and hair color. She's not able to give you grandpa's hair color and grandma's eye color. When she gives you a chromosome 1, you're getting both of those from one parent. They are not assorting independently, those genes that are on the same chromosome. And this is the principle of genetic linkage, okay? So things that are in the same area, especially if they're particularly close on a chromosome, will exhibit high levels of what we call genetic linkage. And this is something we'll talk about a great deal more later in the course. Okay, so we've just spoken about how there can be multiple alleles for the same gene, more than 2, how these things can be inherited either independently or they might be inherited as a linkage group, a linked group of genes. Now I want to speak to you about how what we perceive to be a single trait might be influenced by more than one gene. Okay, so we've used simple examples where gene X controls the color of the flowers of the pea plant, and they're either white or purple depending on the genotype of the individual based on the alleles that they have. Now, some traits, for example, the hair in these dogs is controlled by multiple genes, and there are multiple alleles for each of those genes. And so the combination of the alleles for these different genes is quite important in the expression. So for example, this wiry texture of the Schnauzer on the left is due to this RSPO2 gene, okay, which results in fast hair growth around the face, right? So they have these crazy eyebrows and whiskers and mustaches and beards. And they also have this kind of coarse, wiry hair. Whereas there's another gene that controls long hair, FGF5. So things like Pomeranians that have really long hair have certain alleles for FGF5. And so the genotype for both of these alleles is what determines, is the hair wiry or not, right? So the Schnauzer definitely has wiry hair. That Pomeranian does not. And then how long is the hair? Well, Schnauzer's hair doesn't get quite Pomeranian long because they have different alleles for FGF5, okay? So this is an example where multiple genes are contributing to the expression of a single trait. And we're going to dig into this a good bit more here shortly. Now, one of the things that I want to point out is that just because two organisms share a phenotypic trait doesn't necessarily mean that they have the same genotype. Okay? And that's because there are multiple genes that contribute to the phenotype. There are multiple mutations creating more than two alleles for each gene. And so two organisms might have the same phenotype, like these two snakes, these two albino boa constrictors, could have the same genotype because of different mutations. Okay? Now, they both have a mutation that has led to them not producing melanin. So when you see albino mice, when you see albino snakes, you see albino rabbits, all of these organisms are not producing melanin, okay? And so they have this light coloration, and this is quite characteristic of albinos, which appear in many species. Clearly, this albino phenotype has arisen multiple times in different species, and what is being broken, the gene that is being mutated and no longer functional. So that's a type of mutation, not just producing a different phenotype, but actually breaking and making the gene breaking the gene and making the gene product non-viable. Okay, so these snakes try to produce melanin and it doesn't work. They don't make melanin that makes their skin darker. Okay, and so what we have is a scenario where there's a long molecular pathway, a complex path of events that have to take place in order for melanin to be produced. Both of these snakes did not get to the end point of having functional melanin, but we don't know if they broke that pathway. They broke that means of producing melanin in the same way. And in order to do that, to know if they did do it in the same way, we perform what is called a complementation analysis. And this is a means of determining whether two mutants have the same phenotype because of a mutation to the same gene. Okay? So we know from our previous discussions that this albino phenotype is only apparent, only observed in homozygous recessive individuals. And this is pretty consistent across all instances of albinism in snakes, in rabbits, in mice, in all species. Okay? So these two individuals, we know their genotypes. They are homozygous recessive. But what we don't know is whether they are homozygous recessive for a mutation to the same gene. Okay? Because there are many genes involved in the production of melanin, what if they broke two different genes? They have a mutation that invalidates the product. of two different genes participating in that melanin production pathway. And so a complementation analysis is a simple cross where we take these two snakes and breed them to one another. And this actually happened. So I gave you the example of, these albino boa constrictors selling for a lot of money. Well, there had to be a first one, and those first ones were discovered in the rainforests of South America. So somebody was walking around in the jungle somewhere and found this yellow-white snake, and they sold it to somebody in the United States for a whole lot of money, and that person started breeding them and selling the albino babies for loads of money. And then another one was discovered, okay? And the same thing happened. So now we have two different lines of albinos present in the United States being bred and their offspring sold for goo gobs of money. But we don't know if they are albino for the same reason. And so somebody actually ran a complementation analysis, bred the two albinos from these two different strains, and lo and behold, their offspring were 100% normal in their coloration. Okay, so we know that both of those parents are homozygous recessive, but all the offspring they produce when bred to one another have a normal phenotype. This is an example of complementation occurring. And so I'm going to show you an image from the text, a fruit fly example that makes clear how this happens. So here we've got two different cases of fruit flies with no wings. Okay, and there are two genes that contribute to the formation of wings, gene 1 and gene 2. If you have a mutation to either one of these and you're homozygous for that mutation that breaks the gene product, you won't have wings. So you can see in case 1 that we have the parent on the left is homozygous for a mutation to gene 1, and the parent on the right is homozygous for a mutation to gene 2. In both cases, those parents are then wingless, okay? But when you cross those two and you produce offspring that are heterozygous for the mutation to gene 1 and heterozygous for the mutation to gene 2, what this means is they have a single functional copy of gene 2 from one of their parents. They have a single functional copy of gene 1 from the other parent. See that plus sign indicating the wild type? What we have here is complementation occurred. They have complementary mutations and functional copies of the gene such that when put together in their F1 offspring, the F1 offspring actually have wings. In case 2 on the right, you can see that they do have different mutations, but those different mutations are to the same gene. And so their F1 offspring, while they have two different alleles for gene 1, neither of those alleles produces a functional product. And so they still do not have a functional gene 1 product, and therefore there is no complementation, and the flies do not develop wings. Okay? So both of these are possible. And this illustrates the complexity of the traits that are controlled by multiple genes and the influence that mutations might have on each of those loci. Okay, so the next term I want to introduce you to is pleiotropy. And this is a situation in which we're not dealing with a scenario where multiple genes contribute to one gene product or influence the same thing, or all are involved in this one gene product. Instead of that, what pleiotropy is referring to is when one gene has multiple effects. It affects many aspects of the phenotype. Okay? And so a good example of this is the mutation or the mutations in humans that lead to the phenotype known as Marfan syndrome. So these mutations reduce connective tissue, okay? And it is a dominant mutation. So even if you're heterozygous, you're going to see some of these effects. But the important part is this gene influences many things. One gene, multiple results. So when you have this mutation that reduces the amount of connective tissue the individual produces, It has effects on the strength of the individual's heart, right? There's a lot of connective tissue in that heart, making it elastic, okay? It affects bone length. If you don't have a lot of connective tissue, as you grow, there's less restraint or constraint on your bones getting longer, right? Your muscles and tendons kind of hold that extension back. And so people with Marfan syndrome are very tall and lanky. They have very long bones. Uh, the lack of connective tissue also affects the stability of the lens within your eye, right? It's kind of suspended in place by connective tissue. If you don't have much connective tissue there, that lens can become dislodged. So there are many results of this one mutation phenotypically. Okay, so these are pleiotropic effects. All right. Now the next term I want to introduce you to is the principle or the term epistasis. Now this is when more than one gene contributes to the phenotype for a single trait. Okay? So we definitely observed that in that fruit fly example I used in the complementation analysis, right? Multiple genes are contributing to whether or not there are wings. Here's an example that you are probably more familiar with, at least in terms of the organism and their phenotype. These are Labrador retrievers, exceptionally common dogs, and I imagine at least one of you might even have one of these. Okay? And they come in a few different flavors, the most common being black, chocolate, and yellow labs. Okay? Now, what's interesting is the color of the hair of these dogs is influenced by two different genes. Okay? One of those genes influences the color of the pigment that goes in the hair. And the alleles are big B, which creates black pigment, or small b, lowercase b, little b. That creates more of a chocolate color. So the two colors are either black or chocolate. And your genotype for that, you can be homozygous for black, you can be homozygous for chocolate, or you can be heterozygous, big B and little b, right? And if you're heterozygous, that big B is dominant. You're going to get that black coloration. So how do we get the yellow lab? Well, the other gene involved here has nothing to do with what color pigment is produced. That is the role of gene B. Gene E is responsible for taking whatever color is produced and putting it in the hair. Okay? And so you get yellow labs when you are homozygous recessive for this. That's the only way you get a yellow lab, is you're homozygous recessive for E. And when you have two copies of the recessive E, it means your E gene doesn't work. No matter what color is produced, whatever color pigment is produced by the B gene, your E gene, the product of that, does not take it and put it in the hair. No black or no chocolate pigment is deposited in the hair, and therefore you get dogs that have no color in their hair. They're therefore yellow. Okay? So you can see, look at the genotypes below here, and then also consider this larger Punnett square, right? This is this is like the dihybrid cross that we saw. You can see here the different genotypes and the phenotype that they would produce. Notice that all four yellow labs, doesn't matter what their genotype is for B, they all are homozygous for E. Notice the brown, or what we call chocolate labs, they have a dominant copy of the E gene. Doesn't matter if they're homozygous or heterozygous for it, they have a functional copy that will put their brown color in the fur. But they have to be homozygous recessive for B, right? If you've got a big B, it's black pigment that is being made, okay? So this is an example of epistasis, or the interaction of multiple genes in a single phenotypic trait. All right. Now to this point, in Chapter 3 and thus far in Chapter 4, we have spoken only of what we call autosomal traits. Now, autosomal traits are traits whose genes are found on non-sex determining chromosomes. So for humans, that would be traits that are controlled by genes that are found on chromosomes 1 through 22. Okay. Now, sex-linked traits, in this case, this slide is referring to X-linked traits, so traits that are found on the X chromosome. Every one of you has an X chromosome. I don't care what your biological sex is, you've got at least one. If you're biologically female, you have 2 X chromosomes. If you're biologically male, you have a single X chromosome. Okay? X-linked traits are traits that are controlled by genes found on X's. So females have 2 copies of this and males have 1 in humans. Now, as you see here, the first bullet point, the heterogametic sex. Now in Drosophila and in humans, those are males, right? I'm biologically male. I have an X chromosome and a Y chromosome. Those two are different, hetero. So I am a representative of the heterogametic sex. So some of the early examples of X-linked traits were observed in Drosophila, fruit flies, and the white-eyed trait is controlled by an X-linked gene. Okay. Now the white eye is recessive, and the pattern of inheritance of this white-eyed trait is entirely dependent on which parent carries the white alleles. Okay, and which parent exhibits that phenotype. The ratios are not going to be the same as a normal monohybrid cross. Okay, or even a codominant or incomplete dominant trait. Each of the following slides that I use illustrating the inheritance of sex-linked traits is going to give you some clue as to how these are inherited, and I encourage you to work through some Punnett squares just to make sure that this works for you. Okay. So one other term I want to introduce to you is hemizygous. So males have only 1 copy of the X chromosome. And so I, as a biological male, cannot be homozygous for an X-linked trait. I can't be heterozygous, right? In order to be one of those genotypes, I have to have 2 copies of a gene. Because I have only 1 X, I have only 1 copy of every gene. Thus, the heterogametic sex individuals are referred to as being hemizygous. Okay. Now, I also want to point out that it is not always the case that males are the heterogametic sex and females are not. For example, in birds, it's completely the opposite. Females are heterogametic, males are the homogametic sex. Okay, so let's look at this example of the inheritance of white eyes, an X-linked trait, in Drosophila. You can see here at the top, we've got a true breeding white-eyed female. She's homozygous for the white allele, indicated by those 2 white bars on each of her X chromosomes. Now the male on the right, he is hemizygous, right? That's the term we just learned, for red eyes. He has 1 copy of the red-eyed allele. Note his other chromosome, his Y chromosome, that yellow hooked thing on the right there, does not have a copy of this gene. It's a completely different chromosome. Okay, so you do a Punnett square with this. It's probably best to do it as they did here by drawing the chromosomes with the alleles on them. You can see that when this female produces eggs, in every egg that she produces, she's going to put either the top chromosome or the bottom one, each of them having a white copy of this eye color gene. So she gives that to all of her offspring. Every offspring receives a white eye color allele from the mother. Now the father, the 2 different columns, is a different story, okay? He's going to either give his offspring his X chromosome, and therefore the red allele, or the Y chromosome, which doesn't have any of those. Note that this is where the gender is determined, or the biological sex. The ones on the left column, they receive a red allele, and they are going to be female. Those on the right are going to be male. They are hemizygous. They have only the white allele, and that's what they are going to express. All right. In this example, we're looking at two different X-linked traits. In one case, the X-linked trait is recessive, and in the other, the X-linked trait is dominant. Now, in both cases, we have the mother being heterozygous. Now, you can see it makes a big difference in her phenotype whether if the trait is, or the allele is recessive or dominant. On the left, when the X-linked trait is recessive, the mother is what we call a carrier. She is heterozygous, she has only one copy, it is recessive, so she doesn't exhibit the trait. That's why she's kind of half blue and half white. She's a carrier, she carries the allele, but you wouldn't know it to look at her, right? She does not exhibit the trait. On the right, the mother is also heterozygous, but she is affected. You can tell by looking at her that she has this allele, okay, even though she's only heterozygous. That's because it's a dominant trait. Now let's look at how these alleles are passed to the offspring and what this means in terms of whether the offspring exhibit this recessive phenotype or this X-linked phenotype or not. Now on the left, when the allele is recessive, she passes this white allele to half of her offspring, right? The other half, by chance, get the dominant allele, the blue X, okay? So the two on the left got the blue X, the two on the right got the white X. And dad, of course, gives half his offspring his X with the blue version of the gene and half his offspring the Y. Now, if you look at the right, you'll see that in this situation, the half of the sons are going to have the phenotype. They are going to exhibit this recessive phenotype, even though they only have one copy of the gene. And that's exactly why they exhibit the phenotype, because they have only one copy, okay? His sister next to him, that is heterozygous, she also has one copy of the gene, but like her mother, because she has two X's and the trait is recessive, she exhibits the phenotype of the dominant allele and is therefore not affected. Conversely, when the X-linked trait is dominant, you will see that we have basically the same genotypes in the offspring. Half of the offspring have only the blue allele, so one of the sons, one of the daughters, and half of the offspring have one of the affected or white alleles, those two in the middle. But unlike the example on the left, the daughter here is affected. She exhibits the phenotype, again, just like the mother, because despite the fact that they are both heterozygous, they both exhibit the trait because it is dominant. All right. Now, here's a great example of an X-linked trait in humans, and this is color blindness, which most commonly appears in males. Okay? So it's an allele or a gene that is present on the X chromosome. Now you understand why mostly males are colorblind. It's quite rare for females to be colorblind. And if we look at this pedigree, we start out with two parents. The male is not colorblind. The female is. I'm telling you that this is an X-linked trait. It is a recessive allele, okay? Now, can you tell me the genotype of those two parents in the top row, generation 1? You should be able to tell me their genotypes. That male does not have color blindness, okay? The square, the empty square. And so because he doesn't have color blindness, we know that he is hemizygous for the dominant allele, the normal color vision allele. Now the mother, the blue circle there, she is colorblind. It's an X-linked recessive allele and trait. And so she necessarily is homozygous recessive for the color blindness allele. All right. Now, what does that mean for their offspring? Well, the father is going to give a Y chromosome to his sons and a normal X to all of his daughters. The mother, though, is going to give to every one of her offspring an X chromosome that has the recessive colorblind allele, because that's all she has. Both of hers. She's homozygous recessive for this, remember? And so what you will see is all of the sons are colorblind. And there is no way for her to produce a son that is not colorblind because of her homozygous genotype. None of her daughters will be colorblind, but guess what? Each one of her daughters is heterozygous. Okay? And as you can see down in the third generation, one of her daughters, number 5 there, had a son that is colorblind. That's because that daughter, number 5 there, is heterozygous. She has to be. Her mother, number 2 in the first generation, gave her the recessive allele. And you would expect half of her sons to have the colorblind phenotype. Okay? So I'd work through some of these examples of sex-linked inheritance at the back of the chapter. And you can even Google and search for examples of sex-linked trait questions for genetics. And there's loads of them online that will be helpful in ensuring that you understand. Now, 2 last things I want to touch on. And unlike what I was just talking about, sex-linked traits, there are 2 other categories of traits that are linked to the sex of an individual. And these are sex-limited and sex-influenced. So sex-limited traits are genes that are found in the autosomes. They're not on the X or the Y in humans or in the Z or the Y in birds. They are on one of the autosomes, one of the non-sex determining chromosomes. But they are expressed differently in different sexes. So a great example of this are the feathers of male and female chickens. You can see down here the rooster and the hen. They have very different patterns of feathering, right? Roosters have long tail feathers, and they have this kind of large mane or neck beard that they use when they're displaying to females. The growth of these feathers is controlled by genes not on sex chromosomes, but on the autosomes. It's simply the hormonal influence of the expression of these genes that results in there being different phenotypes, even though those 2 individuals might have the same genotypes. Okay? So that's sex limited. And then there are also sex influenced traits. Now, these are traits that are influenced by the sex of the individual. Okay? So in this case, male pattern baldness, right? So what's interesting here is that different genders can have the same genotype, but they have different phenotypes that are influenced by their biological sex. Okay? So unfortunately for men, male pattern baldness is a dominant allele. So being bald is the dominant allele. So homozygous dominant males are bald. Heterozygous males are also bald. The only people that don't have this are homozygous recessive individuals. Okay? Now what's interesting is for females, the phenotypes are quite different, even though they have the same genotype for this autosomal gene. And the only females that suffer hair loss are those that are homozygous dominant. And even then, it's not as pronounced as this poor fellow on the right here with the sweet comb over. Okay? The influence of this genotype on women is reduced because of the influence of other genes and hormones that are produced. Okay? All right, so this wraps up our discussion of the inheritance of traits and gene alleles that are not necessarily strictly Mendelian, or their expression isn't as we would expect given simple dominance and recessiveness and the Mendelian patterns we discussed in chapter 3. All right.